Hello and welcome to Trovomics. We are excited to have you here using our service and hope that you find some great benefit from it. So in today's tutorial, we will be going over how to create an experiment and correctly upload your FASTQ files and your metadata table for that experiment. So let's go ahead and get started there. We will start on our main dashboard here, where we see our recent analyses, some experiments down below those, but we're going to navigate up to the Create Experiment button. We'll get a nice little pop-up here telling us that our samples need to be sequenced together and not separately. If you don't know that information, you can reach out to your sequencing company to find that out, and they would be able to provide you with an answer. So we'll just go ahead and click I understand. And that will bring us to the first page of our experiment creation, which is experiment details. We'll give this experiment a name of just demo experiment. And we'll follow that up with a description of demo experiment as well. You can get as in-depth with these descriptions as you want, but for simplicity, I'm just going to call it demo. This particular experiment was run on mice samples, so mus musculus, but we have a couple other species in there right now. And our sequencing type was RNA-Seq. So we will hit save and continue. That brings us to our next page with a pop-up as well, detailing information we will need to accurately analyze the data. This information, if you don't know it, we provide an email template for you to ask your sequencing company. So we will click Got It, and we can move through and enter this data. So we have mRNA, we have a poly-A selection method. We used Illumina TrueSeq adapters by Illumina for this specific analysis. The platform model was Illumina NextSeq 2000. We had a paired end read type, so two files per sample, and our sequencing sense is unstranded. So we're going to review all that and make sure it was entered correctly, and we will save and continue. That will bring us to the page where we actually upload our metadata table and our FASTQ files. So if I were to download the experimental design template to take a look at what it looks like, we'll bring that up. And here is our template. So you see our first column is sample name and our second one is file name. These two column titles, we don't want to switch the names of those because that helps us map exactly what we need. Everything else past that, we'll be able to alter as we need, except again, organism, strandedness, library layout. Those should match with what you entered before. We will be able to change things like disease or age and add metadata categories as well. So if you have treatment or sex, you can add those as column titles and then fill out all the information below. So I will show you what a completed metadata table for this particular experiment would look like. And here we have it. So again, we have sample name, np-ang1, matching with my file name for that. We have a sex, a condition, an organism, a strandedness, a library layout, which actually should be unstranded. So let me change that there, and I will drag that all the way through the table. Okay, that's better. And we also have a treatment and a group name. So I will go ahead and save this, and now we can go and upload it to Trovomics. So I select upload, select the file, and now it's there. And now I can upload my sample files as well. So I will navigate to where I have those stored. And I will go ahead and select all of these files. And now they are all there. I have 24 files for 12 samples. So in that metadata table, I had to put the same sample name twice to match with both file names, read1 and read2. So now once I continue, I'll get this pop-up of create samples, check, get pre-signed URLs, check, and upload files with a loading wheel, as well as an indicator of how many files have been uploaded. So we don't want to navigate away from this until it's complete. So once we're done, I will see you guys in the next one. All right, welcome back. Our files have finished uploading. Took about 15-20 minutes for 24 samples. 24 files, 12 samples, sorry. So at the top here, we can see a summary of what we have. We have a condition, a group name, as well as a treatment down below as well. And we can see on the side how many samples it was assigned to. And then we can go in and review individual samples as well. So I can look at MP-ANG1 and see that both files were associated with that sample with the correct lane number and read number. And I could go through all of these samples to make sure, but everything should match up fine and the nomenclature should be correct. So I can save and continue from here, and I will get to a experiment review where I can see my experiment details, that first page we filled out, sequencing details, that second page, and then sample details, that third and fourth page we were on. So again, 12 samples, 24 files. That matches exactly with my files per sample of 2. So once I've created that, I can go ahead and find it on my dashboard. Where did it end up? Right there, demo experiment. And I get an overview of my experiment, a sample summary. I can manage my samples if I wanted to, but we'll get to that in another tutorial. So in the next one, we will be going through how to create an analysis with your newly created experiment. So I will see you guys there. Hope you enjoyed this tutorial. Happy sequencing.